Okay folks, welcome to National 4 and 5 cells and stem cells. Now in this topic we cover National 4, Key Area 8, which is cell biology, controversial techniques. What we are going to do is investigate and debate the use of stem cells. So how we're going to start is we're going to look at what all organisms are made of. And organisms are made up of units called cells. Now cells are the building blocks of life. All living things are made up of cells. Now cells are microscopic, so that means we need to use a microscope in order to see them, and we use stains such as iodine to help us see the cells more clearly. So cells are the smallest structure that are able to lead an independent life, which means they can live all by themselves. One cell can live all by itself and perform all the functions required for life. Now what we're going to spend a lot of time doing today is looking at animal and plant cells and the parts within animal and plant cells and yeast and bacteria and what jobs these parts do. So on the left we have an animal cell and on the right we have a plant cell. So I'm going to go through the structures and their functions. So common to both animal and plant cells, we have a cytoplasm. Now the cytoplasm is a jelly of the cell. It's jelly-like structure, and that's where all the chemical reactions happen. And chemical reactions are required for life. So all the reactions happen in the cytoplasm, the jelly-like structure of the cell. Then we have a nucleus. Now the nucleus is common to both animal and plant cells, and a nucleus controls the cell's activities, for example, metabolism. Ribosomes are spherical structures in the cells, sometimes attached to a structure called an ER, but you're looking for a sphere, a spherical structure in the cytoplasm. And they're very important because they're the site of protein synthesis. That's where all the proteins are made in your cells. Now, we need proteins because proteins are part of the structure of cells. Our antibodies are made of proteins. They help us fight disease. Our hormones, which are chemical messengers that tell our body what to do, and our enzymes, which are involved in chemical reactions. So ribosomes are where proteins are made. We also have mitochondria, which are common to plant and animal cells. And I always think they look like teeth. They look like a structure with three teeth. And if you think about association, mitochondria are where energy is made in the cell. Think you get energy from your food, you eat your food. Teeth, if we think teeth, energy. Okay, so mitochondria are the site of energy production in plant and animal cells. And cell membrane, again, is common to both cells, and a cell membrane is the skin of the cell, and it allows small molecules to enter and exit. Now, as I said, plant cells have all of these structures, but they have three additional structures. They have a chloroplast. Now, chloroplasts are where they make their own food, where they produce their own food. And you sometimes find plant cells called producers because they produce their own food from scratch. And to do that, they need chloroplasts because chloroplasts have a pigment inside them called chlorophyll that can trap light energy, and that light energy can be changed to the energy stored in food. Plant cells also have a vacuole, which is like a bag full of cell sap, salts, sugars and water. And they also have a cell wall, which provides support and structure to the cell. So when you're thinking about animal and plant cells, you need to know the 5 structures that are common to plant and animal cells and the 3 additional structures that plant cells have. Now we're going to look at yeast. Now yeast is a type of fungus, and it has structures common to animal and plant cells. Can you see structures that I would find in both animal and plant cells in the yeast? So what we should see is the cytoplasm, the ribosome, the mitochondria, the nucleus and the cell membrane are structures we'd find in plant and animal cells. Can you see a structure that's only found in a plant cell? Well done, the cell wall. So a yeast cell has all structures that both plants and animals have in terms of the two together. So we have cytoplasm, ribosome, mitochondria, nucleus, cell membrane, all these structures, that has one additional structure that a plant cell has on this diagram, which is a cell wall. Now, not included in this diagram, you will find that yeast cells also have a vacuole. Can you remember what the vacuole was? It was a structure that holds cell sap. So a yeast cell, if you think about all the structures that an animal cell has, and then two additional structures that the plant cell has, the cell wall, which provides support, and the vacuole that contains the cell sap. And we can only see the cell wall in this diagram. And that's a yeast cell ultrastructure. What therefore does the yeast cell lack that other plant cells may have? Well done, chloroplasts. We don't see chloroplasts. We don't see chloroplasts in yeast cells. Now we're going to look at bacteria. Now, bacteria are primitive cells. Can you pick out structures that we find in both animal and plant cells together? Well done. So animal and plant cells, the ones that they have in common here, we've got ribosome, cell membrane and cytoplasm. So you'll find them in both animal and plant cells. Can you see an additional structure that I'd only find in a plant cell? Well done, the cell wall, which offers support. Can you see anything missing? Let's start with something missing that I'd only find in animal cells and plant cells combined. What do I find in both animal and plant cells that is not here? So we should have gathered there's no nucleus here. There's no nucleus, and that's a structure I would find in both plant and animal cells. And there's no mitochondria, which is energy, site of energy production. Can you see a structure that's not in the bacteria cell that I would find just in plant cells? So we don't tend to find a vacuole, and we don't find chloroplasts. Now, that doesn't mean that bacteria cells can't be producers, they can't make their own food. They tend to just have the pigment, but they don't have the chloroplasts. So let's have a closer look at this. In a bacteria, what you find is a cell wall, a cell membrane, doesn't have a nucleus, doesn't have mitochondria, but what you do find are ribosomes and cytoplasm. Now, how does it control cells' activities? Well, bacteria cells have a plasmid. It's a circular DNA structure that allows it to do just that. Okay, folks, we're going to go through the different organelles, where I'd find them, and what their job is. So let's start with the nucleus. What job does a nucleus do? We should have, it controls cells' activities. And where would I find a nucleus? What cells? We've looked at animal, plant, fungus and bacteria. Remember, fungus is yeast. Good, we find a nucleus in animal cells, plant cells and yeast cells. Bacteria doesn't have a nucleus. What does bacteria have instead of a nucleus? A plasmid. So cytoplasm, what does that do? The site of chemical reactions. And where do we find cytoplasm? What cells have cytoplasm? So we should know that it's all cells. All cells have cytoplasm. All cells require chemical reactions to happen. What does a cell membrane do? So cell membrane controls what enters and exits the cell. Only small molecules can enter and exit the cell. What cells have a cell membrane? All cells. Now let's think about a mitochondria. Remember that was the structure with tiny teeth that looks like teeth. What was the job of the mitochondria? What was the association with teeth? Energy. Teeth, food, energy. So mitochondria are the site of energy production. What cells do we find mitochondria in? Remember we've looked at animal cells, plant cells, yeast cells and bacteria cells. All cells except bacteria. Ribosome. What happens at the ribosome? What's the job of the ribosome? That's where proteins are produced. Proteins such as enzymes for chemical reactions, hormones, structural proteins, antibodies. So what type of cell has ribosomes? So we should have all cells. So far, cytoplasm, cell membrane and ribosome are common to all of the cells that we've learnt. Cell wall, what does the cell wall do? Well done, that offers structure and support. And what cells have the cell wall? Think about our cells: animal cells, plant cells, yeast cells, bacteria cells. So we should have all cells except animal cells have a cell wall. And what does a cell wall do? Again, it provides structure and support to the cell. Vacuole, what's the job of the vacuole? Now the vacuole is like a wee carrier bag, if you like. It's a bag and it stores cell sap and, for example, salt, sugars, liquids. What do cells have a vacuole? So in this case, plant cells have a vacuole, and the other cell that had two parts out of the three parts that plant cells have was the yeast cells. So yeast cells, remember, that had everything that was common to both animal and plant, but two additional features that we only found in plant cells, which was the cell wall and the vacuole. And chloroplasts, what cells have chloroplasts? And we should know that the only cell that has chloroplasts are plant cells. Now this is an activity again that you'll do in class, so you'll be able to tell your teacher what job each of these organelles does and where we find them. So let's do this kind of fast. Nucleus. What does a nucleus do? Control cells' activities. And where do we find a nucleus? Do we find them in an animal cell? We do. Plant cell? We do. Bacteria? No, we don't. Too primitive. Fungus, which is yeast? We do. Cytoplasm. Where do we find the cytoplasm? Yes, we do find it in animal and plant and bacteria and fungus. What did the cytoplasm do? It's a jelly-like substance, like plasm, jelly. It is the site of chemical reactions. Cell membrane. What's the job of the cell membrane? Controls what? Enters and exits the cell. So, do we find that in an animal cell? Of course we do. Plant? Yes, we do. Bacteria, of course, and fungus, of course. Yeast has that too. So, again, cytoplasm and cell membrane we find in all of our cells. A vacuole, what does that store? Cell sap. Do we have it in animal cells? No, we don't. Plant cells, yes. Bacteria? No, it's too primitive. We don't find a lot of structures in the bacteria cell. And fungus? We do. So yeast, we do find vacuoles. So cell wall, what was the cell wall for? Structure and support. Do animal cells have a cell wall? No, they don't. Plant cells, yes, they do. Bacteria, yes, they do. And fungus, yeast cells do as well. Remember, yeast cells have everything common to animal cells and plant cells together, but with an extra 2 features that we just find in plant cells, and that would be cell wall and vacuole. So chloroplasts, do we have chloroplasts? Remember, chloroplasts are the site of photosynthesis. We don't have chloroplasts in animal cells. Animal cells do not have chloroplasts. We are not producers. We don't make our own food or produce our own food. Plant cells, correct. Bacteria, no, we don't find chloroplasts in bacteria. Bacteria can have chlorophyll. They can be producers, but we don't find them in that structure. And fungus, yeast, did yeast have chlorophyll? Did it have a chloroplast? No, they didn't. And mitochondria. Now remember, mitochondria is an odd looking structure, and as I said before, looks like it's got wee teeth. What does that remind you of? Teeth, food, that's the site of energy production. So animal cells, they have them. Plant cells, yes, they have them. Bacteria, no, they're too primitive. Fungus, yeast cells do as well. Ribosomes, what are ribosomes? They're the site of protein synthesis. We find them, they're spherical structures that we find in the cell cytoplasm. So that would be your ribosome there. Where do we find a ribosome? Do animal cells have them? Yes, they do. Plant cells? Yes. Bacteria? Yes. And yeast? Yes. And a plasmid. What was a plasmid again? A plasmid was in place of a structure. Plasmid is a circular ring of DNA, and it's in place of the nucleus, and the only cell that doesn't have a nucleus is the bacteria. So bacteria, instead of a nucleus, have a ring of DNA that controls cell activities called a plasmid. So let's have a wee look at the summary here. We can see that all cells have cytoplasm, cell membrane and a vacuole, and only plant cells have chloroplasts, and only bacteria cells have plasmids. Now, we talked about unicellular organisms, cells that only exist of one cell, so they can carry out all the functions and are capable of making all the chemicals that they require to survive, and that they are usually bacteria. Now we're going to have a look at cell types in animals, because we've looked at basic cell structures, but cells take on a variety of different shapes and structures in order to do a particular job. And we call that specialisation. So cells become specialised to do a particular job and to do it very well. Multicellular organisms are organisms that are made up of more than one cell. Human beings are an example of that. They're composed of many different cells, and these cells are arranged into groups called tissues. And the tissues work together to perform a function. So if we have a wee look at a few examples here, we've got red blood cells. So red blood cells have taken on a particular structure. They're biconcave in shape, they don't have a nucleus, and their job is to carry oxygen around your body. So their structure suits their function very well. If we look at the bottom left, that is a nerve cell. Now, a nerve cell's job is to transmit impulses, electrical impulses with messages from one part of your body to another. And the nerve cell structure suits its function to be able to do that. It's long in length and able to have electricity passing along the length of it. Now, cells can vary within a tissue. So if we think about blood as a tissue, you have different types of blood cells within that tissue. So we have red blood cells, we have white blood cells and platelets. We've already discussed red blood cells as carrying oxygen around your body. White blood cells, the job of white blood cells is to fight disease. So within a tissue, you can have different cells working together. Cell types in plants. Cell variety is more apparent when comparing different tissues. So here we have two different tissues in plant cells. We have xylem and phloem. And we can see they're very, very specialised groups of cells. Cells don't look all the same. They're very, very different. And if we look at xylem to the left, xylem really is dead, hollow tubes. Very specialised, and the job of xylem is to carry water from the roots to the shoots, and you'll learn more about that later on. And phloem to your right, again, very different looking cells. The job of phloem is to transport sugar around a plant. So we can see the specialisation in cells. So when we're talking about multicellular organisms, we have a hierarchy, if you like. Multicellular organisms have more than one cell, and the cells are organised into groups called tissues. And the tissues are organised into organs, and they're specialised for their function, and they work together to form organ systems. Now, specialisation of cells leads to the formation of a variety of cells, tissues, and organs, which are groups of organs work together, forming systems. So what we have is cells grouped in tissues, working together in organs, and being part of organ systems. And a tissue is a group of specialised cells that perform a particular function. We know we've got different types of cells working together within a tissue. And we call this division of labour. So if we think about it in terms of a hotel, we've got different groups of staff working together. So you might have the reception staff that are trained to do a particular job, they're dressed in a particular uniform. You've got the waiting staff, you've got the catering staff, the housekeeping, for example, you'll have the bar staff. They all are specialised to do a particular job. Now we're going to rethink about an animal and plant cell essay. So if you were going to take notes on what you've learned about animal cells, what kind of points would you make? I'm going to give you a minute to think about this. Okay, so you might want to say that we know that animal cells have a membrane. And what's the job of a membrane? It controls entry and exit of small molecules. We also know that animal cells have a nucleus, which controls cells' activities. They have mitochondria, which are the site of aerobic respiration or energy production, as we can see at this stage. And they have ribosomes, which are the site of protein synthesis. And they have a cytoplasm, the jelly structure, which is the site of chemical reactions. Now what I want you to do is take a piece of paper and try and think about how you would do an essay on plant cells. So what you might have is plant cells also have a membrane which controls entry and exit of small molecules. They have a nucleus to control cells' activities. They have mitochondria, teeth, site of energy production, ribosomes, site of protein synthesis, but they have three additional features. So you might have got chloroplasts, which makes them producers. They produce their own food by photosynthesis, cell wall for support and structure, and a vacuole for storing cell sap. Now what we're going to look at is where all these specialised cells come from. So we've looked at the basic cell, the structure and function of the parts of the cell. We've had a wee look at how cells are very different looking to suit their particular jobs. We're going to have a look at where all these specialised cells come from. And they come from stem cells. Now what a stem cell is an unspecialised cell. So before a cell has specialised, or what we call differentiated, which is changed to take on a particular shape, it's called a stem cell. So the definition of a stem cell is a cell that has not yet become specialised. But stem cells are able to become specialised cells, such as nerve cells, liver cells, blood cells, cardiac cells, which do a particular job because they're shaped a certain way. Now, growth in animals takes place all over the body, and we need to replenish our cells for growth and repair. So basically, stem cells are unspecialised cells that are able to develop into specialised cells to allow us to grow and also repair anything that's become damaged. So let's have a closer look at stem cells. Stem cells are biological cells. They're found in multicellular organisms, and they can differentiate, which means change their shape into specialised cells, and they also self-renew. We need stem cells to be able to make more stem cells. Now, we have two different types of stem cells. We have embryonic and adult stem cells. Now, embryonic stem cells come from the embryo, the start of new life, and adult stem cells really are found throughout our life. We have these stem cells throughout our life. So let's think about where we all began. We all began when the sperm met the egg. So when the nucleus of the sperm fuses with the nucleus of the egg, you produce a cell, and that cell starts to divide into many more cells. And that group of cells is called a blastocyst, which is really an early embryo, and that's full of embryonic stem cells that are very unspecialised at the moment, but will self-renew to make more of themselves and differentiate or specialise into all the different cells that make up you. So here we have sperm meeting the egg, fertilised egg, our blastocyst full of stem cells able to specialise to produce all the cells that you need in your body. So that's embryonic stem cells. Now, adult stem cells are found in certain parts of your body, and they are really for growth and repair. So, for example, the stem cells in your skin will become more skin cells, and stem cells in your bone marrow will differentiate into more blood cells, like red blood cells, white blood cells. And we need these new cells for growth and repair. So what can adult stem cells do? So the stem cells found in our bone marrow can differentiate to form new blood cells. So for example, red blood cells, white blood cells to help us fight disease. So what stem cells are all about? Really, two different jobs stem cells have to do. They have to make copies of themselves to keep the number of stem cells high, and specialise, differentiate to produce the specialised cells that we require in our body, whether it's when we're becoming a human being from the embryo or throughout our life in terms of growth and repair. So why bother with stem cells? Well, stem cell research is carried out in order to test drugs, understand disease and treat patients, and we're going to have a look at that in more detail now. Test drugs. What if, for example, a pharmaceutical company has come up with a drug that can help treat liver disease? Now, under normal circumstances, we might test on animals or do more human trials, but we know that stem cells can become specialised cells. So what if we were to grow liver cells from stem cells and test the drugs on the stem cells in the lab without the need or would reduce need for animal testing and reduced human trials? So what if we use stem cells to grow specialised cells and add the new drugs to the specialised cells? And see what the drugs do, what effect they have on the specialised cells, and it might reduce the need for animal testing. Understand disease. Some diseases are caused by faulty genes, and again what we could do is use stem cells, create faults in stem cells and use stem cells to study the disease, really grow stem cells with faulty genes and work with these cells to see what has gone wrong and how it could be potentially fixed with treatment. And finally, treat patients. So stem cells are used to make new skin for patients with bad burns. So we have skin stem cells. So if you've suffered a bad burn, what if you use stem cells, skin stem cells, to replace the damage that was done to your skin? There's a lot of prospects for stem cells. The future might use stem cells to grow new cells in other parts of the body. Cancer patients gets a new trachea grown from his own stem cells, etc. There's massive potential, including treatments for diabetes and Parkinson's disease. However, there is a catch, and that's where the debate comes in. The ethics of using stem cells. The disadvantage is that some people believe that using stem cells from the embryo is destroying a potential life, because that group of cells would have the potential to become a human being. So the summary here is stem cells is a cell that's unspecialised, that can divide to form more stem cells and also can become a specialised cell. The advantage is that we can discover new treatments for illnesses, but the disadvantage is using embryonic stem cells can destroy a potential life. Now we're going to have a wee look at processing skills in this unit. So when we're looking to measure the size of cells, really what we're looking at here is a field of view, which is if you were looking through a microscope, this is what you may see. So the exam board might ask you to measure the length of one cell, or the average length of one cell. Now, the important figures here that you need to look at are that the length of the field of view is 300 micrometers. So from here to here, it's 300 micrometers. Now, how many cells do you have in that 300 micrometers? 1, 2, 3, 4, 5, 6. So what we're saying is 6 cells are 300 micrometers length. So what would one cell be? Now, is one cell likely to be less or more than that figure? Less. How many times less than 300? It would be 6 times less. So what we're doing is we're dividing 300 by 6 to get the length of just one cell. Let's have a go at this question. So the diagram below shows a group of cells as seen under a microscope. The field of view was 2 millimeters in diameter. Calculate the average length of the cell. So if we're assuming that the length of cells, the length of the field of view is here, and we know that that's 2 millimeters. How many cells do we have within that 2 millimeters? So we have 1, 2, 3, 4, 5 cells. So what we're saying is 5 cells is 2 millimeters in length. Now I don't want to know 5, I want to know 1 cell. Is 1 cell going to be less or more than 2 millimeters? Of course it's going to be less. How many times less than 2 millimeters? It's going to be 5 times less, so we need to divide by 5, which gives us our 0.4. Okay folks, let's try this question. The diagram below shows a group of cells as seen under a microscope. The field of view was 2 millimeters in diameter. Calculate the average width of the cells. Now if we're assuming the width is this way, again that's 2 millimeters. Either way it's 2 millimeters. Now how many cells have we got across the width of our field of view? So we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So again, we've got a situation where we're saying that 10 cells this time is 2 millimeters. Now, really we're looking for 1 cell. Is 1 cell going to be more or less than 2 millimeters? It's going to be less. How many times less than 2 millimeters? It's going to be 10 times less, because we'll get 10 cells. That'll bring us down to what the width of one cell would be, which is 0.2. Now we're going to look at changing millimetres to micrometres. Now if you have a wee look at millimetres on a ruler, it's very small measurement. Now within that 1 millimetre, if you look at it, you have 1,000 micrometres in there. Now the reason we're looking at micrometres is because we're looking at cells and they're microscopic. They're so small, you need a microscope to see them, so we'd naturally be using micrometres to measure the length and width of cells. And 1 millimetre has 1,000 micrometres in it. So to convert millimetres to micrometres, you multiply by 1,000. And to convert micrometres back to millimetres, you need to divide by 1,000 because there's 1,000 more micrometres in millimetre. Okay folks, we're going to have a wee go at this question now. So the diagram below shows a group of human cheek cells as seen under a microscope. The actual diameter of the field of view was 2 millimetres. And we know that 1 millimetre is 1,000 micrometres. So we have to calculate the average length of the cells in micrometres. So we need to do two things here. We first of all need to find out the average length of a cell, and then we've got to do the conversion from millimetres to micrometres. So the length of the cell, and they've given the arrow to show what they're looking as length, so across the way. How many cells do we have within that 2 millimetres of length? So we've got 1, 2, 3, 4, 5. So we're saying 5 cells is 2 millimetres. So we do the same thing again. We want 1 cell. Is that going to be more or less? It's going to be less. How many times less? Well, it's going to be 5 times less. So we divide the 2 by 5, and that will give us our 0.4. Now let's look at that 0.4 because that's in millimetres. Now I know when I'm looking at a millimetre that there's 1,000 more micrometres in each millimetre. So how do I change 0.4 millimetres to micrometres? I have to multiply that by 1,000 to give us 400 micrometres. Okay folks, the diagram below shows a group of human cheek cells as seen under a microscope. This time the actual diameter of the field of view was 2 millimetres. So calculate the average width of the cells in micrometres. So again, we know that the width is 2, so the field of view is 2 whenever you're going up or down. So how many cells have we got within our field of view? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So what we're saying is 10 cells gives us a diameter of 2, or a width of 2 millimetres. I don't want 10, I want 1. Is it going to be more or less than 2 millimetres, one cell? Less, how many times less? 10 times less because I've got 10 cells. So 2 divided by 10 gives us the 0.2. How do I change the 0.2? Which is millimetres into micrometres. Well, again, I know I've got 1,000 micros in each millimetre, so I need to multiply that by 1,000 to convert millimetres into my micrometres. Remember, there's 1,000 millimetres in 1 micrometre. Now we're going to do some exam cell practice. So the list gives 4 types of cells: bacteria, fungus, animal and plant. Give one structure that is also present in all of these cells. Give you a clue, there's 3 things to choose from. Ribosome, can you think of the other 2? Cell membrane and what's a jelly-like substance? The cytoplasm. Right, which structural feature is found in a plant cell and not in an animal cell? Well done, a cell wall. Which parts of the cell would be found in an animal cell? So what we're looking at here is a plant cell. So what we should have is N and O. Now what is N? Spherical structures in the cytoplasm. Ribosomes. Remember what the ribosomes did? The site of protein synthesis. And what was O? Look at the teeth. Energy production, which is a mitochondria. Right, so A is the cell membrane. What's its job? To control what enters and exits the cell. Well done. What gives shape and support? The cell wall. And what does the nucleus do? Controls cells' activities. So when you go into class, you should be very confident in answering these questions. Now, new cells are produced throughout life. Then cells found in the early stages of an embryo's development, which have the potential to develop into specialised cells. What cells are unspecialised, but they can become specialised? Stem cells. And remember the hierarchy in terms of cell. The cellular hierarchy, you've got cells that are organised into a group of tissues that may find lots of different tissues in, organs, and then you get systems. State a reason other than growth why cells continue to be produced throughout life. Growth and what happens when you break a leg? Repair. Well done. Name a structure present in plant cells which is absent from animal cells. There's a few to choose from. Cell wall, vacuole and chloroplast. Because remember, animals are not producers. They don't produce their own food and you need chloroplast to do that. What name is given to chemicals which are used to make cell structures more visible under a microscope? Stains. An example of a stain would be iodine. Okay, so underline one option in each set of brackets to complete the following sentence correctly. The basic units of living organisms are cells. Most of them can only be seen using a microscope. When examining them, they can be stained, for example, with iodine to make them clearer. So what we're looking at here is yeast. That's one of the key cells we've looked at. So what structure? Now I'm going to change the function here. And I'm going to say support, provide structure and support. So what structure provides support? So what we should have here is A, which is the cell wall. Now let's look at B. What's B? That's the cell membrane. What's the job of the cell membrane? To control what enters and exits the cell. And what controls cell metabolism? Now, cell metabolism is basically one of the cell's activities. So what controls cell's activities? Well done, the nucleus, which is E. Okay, so cell wall would be A. The nucleus is E. Now, I'm going to stop here. There's many more questions that you can have a go at. What I advise you to do is practice the essays, plant cells, animal cells. Have a read over the PowerPoint again, practice the questions so when you come into class, you have learning behind you and the learning will be so much easier for you. Well done.